So this is the second part of the membrane lecture, and we're going to talk about proteins and carbohydrates, the other two major components of the membrane. All right, so what you need to know again is the amphipathic. They do have a hydrophilic and a hydrophobic section that kind of decides which side is which. And there's two different types. There are integral proteins, I need you to know, and those are transmembrane, like you see in the picture here, where they go all the way across. Or they're partially embedded, which means they go in and they might be only halfway through. And then we also have peripheral proteins, and these would be loosely bound to the surface of the membrane. So they're kind of stuck on there, but not really embedded in, and they can be either side. So just realize that both proteins exist and may have different functions. So what you're going to find is this: the proteins, like everything else, has a lot of functions. And I've kind of debated on how much to go in through this, but I think for our purposes, just kind of knowing what they do and making sure that we're good with this. So transport, we talked about this before. They can act as channels. Here you can see a channel and actually active transport may be happening on the right. We're going to talk about that a little bit later. They can act as enzymes. They can be embedded within and act as enzymes. They can be signal molecule receptors where the ligands actually sit in. Cell recognition that we haven't talked about yet, kind of mentioned with the cell communication. But that's in conjunction with, you can see the green, that's with the carbohydrates. The proteins can actually recognize the carbohydrates on another membrane and allow for them to come together. Intercellular connections here. Inter is mean between. So here we have two membranes. It turns out proteins can have kind of a stickiness to them. And so what that'll allow them to do is to stick together. I'll let two membranes actually hold together. And then on the right-hand side, we actually have attachment to the cytoskeleton. Again, they are sticky and then allow not just to each other, but also to the cytoskeleton. So that would keep this membrane maybe in place in the cell someplace next to some of the cytoskeleton structures. Okay, so those are the 6 functions that we have for you that I want you to know. And then the carbohydrates. Again, this is really mostly for cell recognition. And we have 2 different types. We have glycoproteins and glycolipids. And like I said, on the old books up until this new edition, they weren't actually identified correctly. I mean, they were explained correctly, but just the picture was wrong. So a glycoprotein is a carbohydrate attached to one of the proteins. And a glycolipid is actually one of the carbohydrates attached to the phospholipid. So it's as easy as that. It's not real hard, but again, the pictures were a little off or deceiving. And again, here's cell recognition. And just to kind of let you, allow you to understand how we're using this maybe to fight other things. Here's HIV, and HIV is known for the CD4 receptors. That's how they get in. Unfortunately, our cells are not smart enough to keep them out. They hook onto the CD4 receptors, and our cell actually brings in HIV. But it turns out they need a co-receptor, and that is the CCR5. And what we're looking at is ways of maybe shutting down those CCR5 receptors. Whereas then when a HIV actually shows up, it looks for recognition. And typically with the 2 receptors, it would recognize it and pull the cell in. This time, without that other receptor, it doesn't know what it is, and then HIV is deterred. So just a way of using cell recognition as a way of fighting some diseases such as HIV, the human receptor virus. So very cool. And then the other thing what they're finding out, and this is not in the old books, but actually this what's outside versus inside. The sidedness, we talked with the Golgi apparatus that we have a cis side and a trans side. And what they're recognizing now is that might be due to the actual carbohydrates that are attached, giving it its sidedness. And so that can be helpful. Again, this is a little bit more detailed than we knew before. So pretty cool stuff. And we'll stop the share. We'll stop the